Thanks very much for inviting me for the introduction. I'm really bad at crediting other people during the talk, so I should probably do it now. This is all work with a bunch of other people, including my previous supervisor, Eamon Gaffney, and also Kenta Ishimoto, who's from Kyoto, amongst some other people at UCL. So today I hope to talk about something you might not have heard of or might not too much about. That's the aim. It's quite vaguely multiscale methods and microswimmer models. A good way to start that off is to probably say what a microswimmer is, which would be helpful if the slide advances. There you go. So this is a microswimmer. This is the canonical microswimmer. It's a sperm cell. So by microswimmer, I mean something that swims on the microscale. So a sperm cell like this is probably 50 microns-ish in length, and will often move and swim at maybe 1 cell length a second-ish, give or take. So very small scales, sort of Stokes flow all around them. Many other examples of microswimmers, and they can all be motivated by either sort of industrial or medical applications. You've probably heard of bacteria, I expect, but this is E. coli in particular, from taken about 20 years ago. And they swim around. They're essentially these spheroidal-like blobs with big helices attached to them, which they rotate very, very rapidly in order to corkscrew their way through fluid. And then one which is a little bit weirder, you might have seen it if you're into your biology a little bit, is this algae, Chlamydomonas, which is this roughly spherical thing that does breaststroke type swimming. I hope you can roughly see that on the screen. So it has these two flagella or cilia, as they are, these thin appendages, and it literally drags itself through the fluid like breaststroke. So when you put lots of bacteria together, you can get quite cool things happening. So this is an example. What you're seeing in that video on the right are individual bacteria. There are just many, many of them in this quite dense suspension. And when they're all together, they can give rise to this weird phenomenon of bacterial turbulence. So this looks roughly like a turbulent flow would in that it's got nice vortices, there's big coherent structures, which is particularly strange because Stokes flow, which governs the fluid dynamics, doesn't admit any such vortices or anything turbulent. If you're nodding and you think that this would be a really good direction for the talk to go, then I'm really sorry, but we're not going to look at anything more than 2 swimmers in this. But there's a huge field of active matter that does this, and if you're interested, it's very, very rich. We're going to focus on these individual type ones. And in particular, we're going to focus on a special property that you can see in each of these videos. And that's these separated timescales, or what I'm going to hopefully convince you are separate. So if you look at the sperm cell in particular, as it's swimming about, it's flicking its tail quite a lot. So the tail is moving quite a long way, and actually quite quickly. But in comparison, if you plot the trajectory of the sperm cell, it's moving an awful lot more slowly than you might expect. It's kind of wasting a lot of effort by moving its flagellum an awful lot, and it's not really moving very far. The same is true for the Chlamydomonas as well. It's like its flagella are moving quite quickly, but then it sort of just drags its body a little bit with each step. So it's taking a very long time to swim. And the same is true for the bacteria. I hope you can see the corkscrewing on this screen, whereas the actual speed that they're going is an awful lot less than the speed that they're rotating their flagella. So the idea is that there are fast timescales of microswimming and slow timescales of microswimming. The fast ones are typically, undulations of a flagellum, whereas the slow ones would be the overall motion and the swimming trajectory. This is quite nicely illustrated by this simulation that I've stolen from someone else of a sperm cell, which is in a background shear flow, which is causing it to rotate a bit. And you can see there's this quite, maybe relatively interesting long time trajectory sweeps out this sort of ellipse type thing. But I hope you can just about see it. The actual sperm cell itself is undergoing some quite fast wiggling, and the frame rate of that's like downsampled by a factor of 10 or something. So this is really oscillating very, very quickly. And typically, we're not really interested in these fast oscillations. We're much more interested in, well, why has it done that very large shape and that particular trajectory? So the hope for this talk is to try and gain access to this sort of information. We won't solve this particular problem for the sperm, but we will hopefully move in the right direction. So I'm going to talk about, for the entire talk, the method of multiple scales, which may be something you're familiar with and you may be entirely unfamiliar with it. So the first sort of third or so will be taking you through what is the method of multiple scales when what I hope is a really simple example that maybe builds up some intuition for this. We'll then look towards a more complicated problem where the mathematics actually gets quite interesting and we even relate it to an experiment and arguably do something useful. And then we'll take it back to a very sort of simple theoretical problem right at the end. And I've pitched this perfectly, so if you know a lot about the method of multiple scales, this will be very patronizing, and if you've never heard of it, it won't be enough detail for you to understand. So I'm going to alienate everybody all in one go. But do bear with me, and do feel free to ask questions at any point. So consider this swimmer and a really simple model of propulsion, which is ignoring basically everything. We're just going to say this swimmer has coordinates x, y, and it moves at some constant speed v in some direction given by phi. Okay, well, that's rather easy, particularly if phi is a constant, then even I can integrate these equations and it just moves on a straight line path. Perfectly easy. The question is an awful lot more difficult if the direction that it's swimming changes with time, right? I certainly can't integrate that in general. So, okay, let's try maybe what is the easiest case that isn't just constant. Let's suppose that it's changing its direction in a sinusoidal way with some amplitude. Hopefully we can make some progress. But unfortunately, I can't solve this, and you'll note there's a key thing that I didn't mention is I'm saying this is going to oscillate quickly. So omega here is some very fast frequency, which will be the fast frequency that the sperm was, say, moving side to side or facing in different directions. So we have this very rapid change in the way it's facing versus what is sort of an order one speed. So the idea is, can we somehow work out how this is going to swim and kind of average out these rapid oscillations? If you solve this numerically, you get the black curve, which indeed is undergoing some sorts of oscillations, but really it's just sort of moving to the right with some oscillations about that. It would be nice if we could actually recover that systematically from the dynamical system rather than having to numerically solve it.
Quick question about your sort of general graph there.
Please,
is that true for all omega, or is it just for omega large?
Is what's true?
Is that general?
Oh, this is, that's a great question. I expect it's definitely true for omega large, because that's how I plotted it. In general, I imagine that is still also true, naturally stretched out a lot. That's actually, I actually never tried, but that's I'm always very focused on something very fast because of the motivation. But certainly the analysis that I will show you will completely not work if omega is not very large. Very much going to exploit this separation of timescales. Indeed, we introduce, as if you've done multiple scales, this will be familiar to you, the fast time scale capital T. So that's basically zooming in on these fast oscillations. So the oscillations happen at order one capital T, which is the same as saying very, very small changes in little t. So if you plug that in, we haven't done anything, we've just changed the label, we swapped the omega t out. But then the sort of magic behind the method of multiple scales, which you kind of have to take on faith unless you really get into the hardcore analysis of it, is to treat these two time variables as formally independent, so independent quantities, which is clearly untrue. But if you treat it like that, you can make some progress. So that converts our ODE, or non-autonomous ODE, into a PDE system, which is arguably not better. It seems a bit more complicated. We've made the problem worse. But we will at least be able to make some progress later on. So omega is very large. This is an asymptotics problem, so you almost always would expand about expanding powers of your small parameter, which here is 1 over omega. And we're really just going to be interested in finding this leading order solution, the x naught and the y naught, which hopefully is going to be a horizontal line for us. So if you take that expansion and you plug it into that PDE, and you look at the highest order terms, so the highest order terms are going to be the ones with an omega in front of them in this, because omega is large. And it turns out the only ones that you pick up are dx naught by d capital T, and the same for y naught. Everything else is order 1, so it doesn't contribute here. So all those equations tell us is that our leading order behavior doesn't depend on the fast timescale explicitly like this. So really the quantity we're looking for should be independent of capital T. That's kind of what we're hoping for. We want a horizontal line at the end of this that isn't oscillating very rapidly. Okay, if you go to the next order, then you now pick up from this term, you pick up the derivatives of this quantity, so the dx naught by dt. And notably, those are proper derivatives now in time because they're independent of capital T. But now this is still a PDE system. We still have dx1 by d capital T, and we know nothing about this next order part. So this is the next bit about multiple scales, which I really, really like, is there are two ways of thinking about this. To make some progress, you can close your PDE system by insisting that your solution is periodic in capital T. So if your solution is periodic in capital T, that means if I was to average this equation over a period, then these two time derivatives vanish, because I'm evaluating it at the same thing at ends of the period. Another way of looking at this, which I actually think is a bit more intuitive, is this is just the Fredholm alternative theorem. So this is just the same as the solvability condition for this linear operator on the x1 and the y1. The solvability condition turns out to be this quantity, this average must turn out to hold. Okay, so if you take that on trust, which I promise does work and is actually really rather neat, I have these effective equations for dx naught bar and dy naught bar. This is our leading order dynamical system. And it turns out that if you're trying to compute the average of cos of a sine, then you can do this explicitly. It's just the definition of the Bessel function, actually, which took us by surprise. My collaborator spent a long time doing some contour integral to be able to compute this average, and then somebody else just pointed out that it was the definition of the Bessel function. So that was a nice research experience for them. The second one is much easier. It just vanishes by parity, so that goes away. So what does this mean? This says that our leading order dynamics, there's nothing in the y direction, which fits with our figure. We didn't have anything going upwards. And in the horizontal direction, we've still got the speed v, but it's being modulated by this Bessel function evaluated at the amplitude. And if you're not acutely familiar with these Bessel functions, this particular one is something that is always of size less than or equal to 1, and it kind of oscillates and gets smaller and smaller in magnitude, and it swaps sign. So in particular, because the speed is v times this Bessel function, it means that if my a is non-zero, then these rapid oscillations are just going to strictly reduce the speed that I was going, which kind of makes sense. You spend some of your time not facing directly to the right, so you're going to go slower than if you were just facing directly to the right. And in particular, if you did some absolutely enormous oscillations, so if you were basically spinning all the way around, then your effective speed is just in the opposite direction. And so you can even reverse what you were doing. So you can get quite big qualitative changes in this quite extreme example. Okay, this was a very easy example. We had a sinusoid. If you take your phi to be somewhat more general and have some maybe slow time varying component, and then capital G is doing the same job as the sine was in the previous example. And then you can repeat all of this analysis and you can go through. It's not terribly exciting. It really is identical. And you get some sort of effective speed out of these equations and then some effective bias angle out of this, all depending on your capital G. You just have to compute a particular average of it. So all this is doing is taking the average of cos G and sine G, right? That's all that's doing in maybe a neat way. And you get exactly the same results from this in that RP will have just averaged e to the I something. So I'm going to get something of size less than 1. So my speed is definitely still reduced. And then I get this potential for bias, this theta p.
Are we still assuming that G is periodic?
Yes, yeah. G definitely periodic. Yeah, that will be true of everything here that's on a fast time scale will always be assumed to be periodic, which it tends to actually be in these microswimming applications. They really are pretty periodic, potentially with some slow drift like I've got built in there. So if you plot this for some particular G, well, the top one's just the sinusoid, which we already saw. And the blue line here is the solution of our leading order system. So the idea is the blue should go through the black, and that's perfect. That means we've done a good job. It does quite a good job for this G, and it predicts that we just go in a straight line. This is maybe a bit more surprising because this isn't a nice function that you would be familiar with. But then for the last one, which somehow looks a bit less symmetric than the other two, you do get some bias angle. Now, I'm not claiming that these results are particularly deep because all we're saying is what is my average heading, or essentially the average vector direction that I'm going in, and just adding that up. So it's not surprising that if the sine of my angle has some non-zero average, then I end up going not in a straight line. This isn't exciting, but it is hopefully slightly intuitive. There's actually quite a nice condition. These two things are something known as anti-periodic, which I'd never heard of before. But it's as if you were to be periodic, but then halfway through your period, you just do the opposite of what you did at the beginning. So it's a symmetry that sine and cosine have, but also that this function has. And that's a sufficient condition to just move in a straight line to the right. Okay, quite a simple basic example. The reason that motivated me to even look at this was, what if I take a nice simple swimmer and I put it in a shear flow? So just like that sperm was, and it was rotating around, what can I do a bit more abstractly? So you might have to take it on faith that this is the correct equation to write down. This is saying that the angle evolves in time with some weird trig function with this parameter b, and then plus this omega g prime of t. Hopefully you can maybe see that that last term is just doing the same job as our capital G of t was doing. You integrate this up in time and you get phi as something plus capital G, so it's sort of entirely analogous. But the rest of the functional form seems a little bit mystical. So this does just come from Stokes fluid mechanics, but it's really rather famous. So if you ignore the omega g prime of t, then this equation is Jeffrey's equation, which is fairly famous. Jeffrey was rather famous, which he wrote down and published, I think, precisely 100 years ago. And this has some really neatly understood solutions. You can actually just analytically solve this thing. And you get these two qualitatively different regimes based on what b is. So b here is a parameter, just a scalar parameter, that captures the shape of the object. So typically for a spheroid, you would get b somewhere between -1 and 1, and as it becomes more and more elongated, it will get closer to ±1. B equals 0 is just a sphere, which makes some sort of sense. If I get rid of this term, then that's saying a sphere in a shear flow, which just causes things to rotate, will just keep rotating at a constant rate. So that's at least a little bit intuitive. So the question is, am I still going to get a Jeffreys orbit out of this thing if I've got this object that would be rotating in a very well-defined way, but now I'm giving it a little kick all the time. It's kind of oscillating rapidly. So you can do the same sort of analysis, and I'll go through it quite quickly. Now you actually pick something up at leading order when you do an expansion, because there's an omega on the right-hand side. And this is precisely why we write this down. It's basically just giving us what we would expect. We say, I want phi to be something slow plus some very rapid oscillations, which would be this g. But we have to determine what this constant of integration, or really function of integration, is. You go to the next order balance, you can write something down, you pick up some stuff. You do the averaging. This averaging is fundamental to multiple scales. So really you should think of everything I'm going to do in this talk as averaging out over fast oscillations, but doing it in a really systematic way. I'm not just sort of naively waving my hands and saying I'm going to average everything. So you can impose periodicity or invoke the Fredholm alternative theorem, and you get this effective equation. So now I have to average cos of 2 phi naught. So I know what phi naught is, at least a little bit, in terms of capital G. So you can just compute this average. If you do it, and you have a nice way of averaging 2g now, or cos of 2g and sine of 2g, then you get this equation out. And if you can remember back 1 or 2 slides, this is exactly Jeffreys' equation, but the parameter that was our shape has been modified by this Ra, and now I've got some sort of phase shift, which is equivalent to the bias that we had before. So what that means is that our object, which is, say, like an ellipsoid, but now it's oscillating very quickly side to side, the effect of those oscillations is to modify the shape that it has in the flow. It's almost like it's taking some average shape. If you were to close your eyes or blur your eyes a little bit and flick side to side, it's what shape is it having? And then plus some little bit of bias. Okay, still rather abstract. Yes, go for it.
Is your RA less than 1 again?
Yes, it is still less than 1. So I've got an e to the I something, which is average. So it's actually quite neat. It reduces the shape. Actually, as I think that comes up on the next slide, this is quite nice. So it strictly reduces the size of this shape constant. So you can get weird and wonderful shapes that have b outside -1, 1. And these are typically very contrived. And Bretherton, who this parameter is named after, has a lovely paper where he describes some of these, but they're quite exotic. But what you can do is, because this RA is always less than 1, it means that if you had something that wasn't oscillating, that would be in one qualitative regime where it tended to a steady state, which is this black line. But then the oscillations drive its effective shape to look something more like a spheroid, which has periodic motion that doesn't tend to a constant. So just a tiny little bit of oscillation can completely change the qualitative dynamics of what's going on. So I think a good takeaway from this is that it can be really important and quite subtle. Like this extra kick that you're giving it might change more than what you might naively expect. I was going to talk a little bit about higher order drift, but it's a little bit less interesting. If you look at the next order terms in the expansions for all this stuff, then you can track the error. You should expect error to accumulate. We've taken a first order thing. You should expect error of sort of 1 over omega. Like that will happen. But if you look at this plot, this is the difference between the true solution as computed from numerics and then the leading order solution. And indeed it grows, it then sort of oscillates a bit and is still growing, but it takes ages to grow. You should expect this to happen on t is order 1 or maybe 1 over omega. And this isn't happening at all. The error is way, way slower in growth. And you can show that if you have this kind of neat condition on the oscillations that you're imposing, then so long as that function is anti-periodic, then it puts the error down another asymptotic order. So actually this asymptotic approximation is really very good, so long as your swimmer does something and then kind of precisely undoes what it did, which is quite nice. It's sufficient rather than necessary, but it's quite easy to show that it's sufficient. Okay, but while we've been talking about this, we started off with real swimmers. So if we take this real swimmer, and this is clearly not a real one, this is a model sperm that is just nicely idealized. Its beat pattern is roughly shown there, and it will do this at some frequency. So we're going to simulate this swimmer in a background shear flow. So the same flow that was causing these particles to rotate. And if you do this for very low frequencies, say omega is 1/2, and then you ramp up omega, taking it through unity, which is weirdly low, and then up to 20, then what this swimmer actually looks like it's doing is its angular dynamics are just following a Jeffreys orbit. And this G, by the way, is measured. So you simulate this sperm cell, and you can measure how its orientation is changing because it's flicking its flagellum backwards and forth. So you can measure G, which looks suspiciously like a sine wave, which is quite irritating when things turn out to look very simple from biology and makes it look like I've cheated.
So that's experimental data.
Let's go for in silico experimental data. Yes. So the waveform has come from experiments. Everything that it's doing in this background flow is simulating that. And because this is all kind of physics driven, we're relatively confident that that is actually roughly what's going on here.
So, I mean, presumably you could actually work out what G gave you the maximum velocity, you know, if you sort of limited the energy involved.
Yeah, that is.
And would it look like a sine wave?
This is actually a really good question. I definitely don't know the answer to, but I know lots of people have looked at it. So for instance, there's a group in Birmingham with Dave Smith, who you may or may not know, who's done a bunch of stuff on optimizing what sort of traveling waves of moment you would put down these flagella to get them to kick. So it's quite a complicated elastohydrodynamic problem. Because unfortunately they're beating response to their environment, which is actually quite irritating from modeling it like this. This certainly didn't do that. This is a prescribed beat. There's lots of more classical stuff about efficiency of sort of Taylor swimming sheets, these kind of infinitely long thin sheets that are approximating flagella. And you can get some nice analytics out of that. But as far as how to actually do this with a swimmer, I think that is beyond the realms of what I can do. Or it's very hard to pose a nice, well-defined question, actually. But it is very interesting.
Could you carry on with the idea that it's being simulated later? You said some.
Oh yeah, for sure.
Could you approximate that to a sine, and then just do it whole thing analytically?
Yeah, could do. Absolutely. You also the stuff you have to do numerically isn't that taxing in particular. Basically, you just need to compute the average of G, or cos G and sine G. So this particular one will basically almost follow a Bessel function. Because it looks so much like sine. But yes, you definitely could in this case. Whether you would is a more philosophical question, but yes, you could do. The important bit is that looked like a Jeffreys orbit. But why did it look like a Jeffreys orbit? That's quite unreasonable. This is a complicated swimmer that looks nothing like a spheroid. It's got this thin tail, it's flicking backwards. But it turns out, and we didn't know this at the time, this was something we looked at later, is that if you take a general shape-deforming swimmer, so say this is like the chlamydomonas we have, so it's all the sperm cell, that deforms its shape in the plane of the flow, which is what the sperm was doing. It was kind of just flicking its tail backwards and forth, or the chlamydomonas doing its breaststroke. It's kind of doing this deformation in the plane. Even though it's 3D, it's deforming in the plane. If you do that and you put it in a shear flow, then as rigorously as this asymptotics can be, you can start from fundamental Stokes equations and show with this asymptotic analysis that the effective equations you get out for the orientation are just exactly the same as those that you would get if it was a rigid body of some effective shape, potentially with some bias as we had before. That's what this theta C is. So this is actually we were really surprised that this kind of fell out. So you take any shape deforming swimmer, as long as it's deforming quickly and periodically, but that's like what the swimmers I showed you were doing, and you put them in a shear flow, they will look like this classical solution of Jeffrey from 100 years ago, which was very surprising to us and obnoxiously neat. It shouldn't have turned out quite so neat, but it's very nice. The obvious question is, does this apply in 3D? And the answer is maybe. The analysis is horrendously more complicated, and it's all basically the difficulty of dealing with Euler angles or all the things associated with problems with doing a much more complicated dynamical system. If you're interested on how to do that, it really is very easy, and there's a publication by him and Gaffney on how to do this, which is included in the links at the end. But it's really rather neat. It's quite short as well. Okay, we maybe have a bit of intuition. I hope you have a little bit of intuition for multiple scales. It's sort of systematically averaging, and you can get some quite neat results that, from the nice dynamical systems you get from Stokes, can give you nice, clean answers. So now what I want to do is talk ever so slightly more briefly about a more complicated problem, which is directly motivated by an experiment, not an experiment I did. So earlier in the year, this paper came out in JFM, which was about a chlamydomonas in a channel. So they're in a channel here, there's flow pushing them that way. So this is a quasi-flow, if you're familiar with flow in a channel. And the chlamydomonas in their experiments always seem to be facing upstream and swimming. This is very hard to see at the resolution of this screen, but you might be able to see them doing their breaststroke in that direction. So they're trying to swim upstream, they're just being affected by the flow a little bit too much to actually move to the left. But this kept happening. It always was collecting at the center of the channel and pointing upstream. And the authors didn't know why. I didn't know why either. Here's the sort of the caricatured picture that they drew, is that you start off with this chlamydomonas and that they eventually end up at the midline facing upstream. So why is that? So they posed a model, they represented their chlamydomonas like this, and it wasn't an ellipsoid, it really did look a bit more like the chlamydomonas. And if you ignore the walls, then you can write down a nice simple dynamical system that's a pretty plausible model of this. And by plausible, I mean it's sort of faithful to the Stokes equations behind it. So it's not just phenomenological, this is actually the systematically derived model for a certain class. So the model is kind of similar to what we've seen before, at least in vein. We have some orientation theta, and this term looks exactly like the Jeffreys equation term. It's basically saying how much is this being rotated by the rotation of the flow that's happening. It's here in this background flow, it's moving faster in the center than at the edge, so it's causing you to rotate like this. And you similarly, you have this sort of swimming speed, but you're only going to track the speed away from the midline of the flow. And you're going to assume that both the swimming speed and the shape oscillate in time. So is that reasonable? I think it is reasonable. So as this thing swims, it changes its shape. So the parameter that describes its shape should change. That's very reasonable. And the swimming speed was definitely not constant all the time. You could see it from the first video. It kind of had a bit of propulsion when it was doing that, and then it slowed down an awful lot. And they can measure these things. So I think they did this from in silico, where they simulate this chlamydomonas moving, and they can compute, apologies for the changing case between the U's, but they can compute the swimming speed and they can compute the effective shape parameter of this chlamydomonas at each point during its beat. And this is periodic. So U and B are known functions. We can prescribe them. We just measure them from our experiment and we say, or from our in silico experiment and say, this is this swimmer. We know U and B. The question is then, is adding in these oscillations enough to recreate this observed behavior that they have, this long time behavior of being in the middle of the flow and facing upstream? And it turns out the key to this is that Y that's popping up in here, because the shear rate of the flow is going to change as you get further away or closer to the midline. When you're at the middle, there's sort of no rotation in either direction, so it vanishes. So we're going to skim through this, but the maths is much more interesting, but it's not obvious that it's more interesting. If you do some numerics and you simulate this system at the top right for omega very large, then somewhat expectedly, you get quite fast oscillations. They're pretty small at the scale of this plot, but you can hopefully see that that's not a smooth curve. It's kind of oscillating a bit. And these are happening on the time scale of order 1 over omega. So that's driven by the oscillations in the U and the B directly. On an intermediate time scale, which is perhaps a bit more surprising, you get these longer oscillations, quite large. You can maybe see that this is still wiggling, so the fast oscillations are still included in this. And this seemed to be happening on a time scale that scaled with root omega. It's not abundantly clear why it should do that, but it seems to. And then if you look at it even longer, for really longer times, which well, arguably are just order 1, then you have this big qualitative shift in behavior that could happen. Like there are these oscillations happening, and then somehow the character of them completely changes. So it looks like this system doesn't have just 2 time scales, it has 3 time scales in it that we need to consider. Okay, so we do that. It's called the method of multiple scales, not the method of 2 scales. So it is capable of handling that. It's just a very natural extension. It's just a bit more complicated. So we have 3 time scales, capital T, which is very fast, tau, which is intermediate, and then little t, which is just a normal experimental time scale, if you like. And just to be really clear, if you're going to transform t into these 3 time variables and treat them as formally independent, your derivative operator transforms like this. It's just simple chain rule, but it's nice to note with at least 3 things.
Do you know there's only 3?
No. So the question is, whenever you see a system like this and you think you should do some multi-scale analysis, what on earth are the relevant scales? Often you can do, as in fact we did to find the next scaling, was you can wave your hands a little bit and say, well, if this changes by an order 1 amount, how much does that change, and so on. Finding this intermediate timescale was not at all obvious, and indeed was surprising that it was even there at all. Go for it.
So, sorry, I'm lagging a bit behind. But with these particular bacteria, they're not oscillating side to side.
They're not oscillating side to side. They're just changing. So they're doing breaststroke. I'm still at the front of a room doing breaststroke. They're doing this, which changes their shape. Sure, it's in an axisymmetric, in a symmetric way, but they are changing their shape. So B changes, and their speed in the straight direction is faster when they're doing this part of the stroke, and then it's actually negative when they're doing the other bit. So that's what's driving the oscillations. There's no rapid side to side as there would be in the sperm, for instance. Does that make sense?
Yep. So the cos term there in
the cosine term there is saying if I have a rigid body in Stokes flow that's aligned at this angle theta, then how quickly is that being rotated? And that will depend on the angle that it makes towards the flow. Notably, that vanishes if B is 0, which is a sphere, because it doesn't matter which way I orient my sphere because it's symmetric. So yeah, the cosine is nothing about oscillation. It's all about these Jeffreys orbits of it matters which way I face the flow. So if you do plug in these different scales, and this is one of those magical transformations that it seems that actually you should rescale your vertical coordinate by root omega. It's not obvious why you should do this. If you try and do the analysis without it, you'll hit a brick wall. You can be a bit careful in doing it. But if you do this scaling, then you end up with a system which just looks a little bit more complicated because some root omegas have appeared in places. I'm not going to go through the gory details of this, but they are very neat. If you expand this in powers of root omega now, root is, when you're doing asymptotics, you never know what expansion you should use. If you see a root omega, you should probably include it in your expansion, and that's what we did, and it seems to work out. But there's always a question of uniqueness. So if you do this, and you do the same thing I've been talking about this whole time of averaging out these fast oscillations, so you average out in capital T in particular, this fastest scale, it turns out that your Z0 and your theta 0 don't depend on capital T for exactly the same reason they didn't in the previous problems. But at the next order, you get this system having averaged. And these averages are just the average speed and the average shape. There's nothing nonlinear going on in terms of these parameters. We've just been able to average them easily. Okay, that's nice. This actually looks a bit like an ODE system. If you treat tau as your time parameter, this just looks like an ODE system. It's quite a nice dynamical system, actually. These things correspond to closed orbits in this phase space. And this was well studied sort of 10 years ago by accident that we stumbled upon the same sort of system. If you take a rigid particle in flow, you get this sort of equation that pops out. And you have nice closed trajectories, and then these are also closed because it's periodic in theta, but quite different character. And these are all captured by a first integral. We were able to borrow this from the study 10 years ago or so, but if you compute this for some not particularly nasty function g, it's just not nice enough to show on a slide, then you can really show that this is conserved throughout the motion. So we should expect h0 to not vary with capital T, not vary with tau, because it's a first integral of this system, but it might plausibly depend on the longer timescale. So the reason that this is interesting is that this diagram completely captures the behaviours that we might expect to see in an experiment. So at the very centre of this diagram, you get upstream swimming. So that corresponds to theta being pi, so that's I'm pointing this way, and my z being 0. So that's kind of the behaviour we're hoping to get out as the answer of our analysis. And then in the opposite regime, you have kind of endless tumblings. This is where I'm always in one half of the flow, and my theta, I just spin around, round and round and round, because the flow's doing that. And then there's this kind of separatrix in the middle. So that begs the question, can we just track h0? That would be great. If we can track h0, see how it evolves in time, that might tell us what the answer is going to be. Okay, so to do this a bit more formally, you really want to start off with something that wasn't part of your asymptotic approximation. So you just remove the noughts from everything. And you can go through the sort of analysis, you expand h sort of trivially, you just get back h0, and do the proper triple scale analysis, you average over all these time scales, and what you get is a nice autonomous ODE for h0. Okay, we have an autonomous ODE, great. That means we can, in theory, work out what's going to happen. We know f explicitly as well. It's a horrible formula, but we know what f is. Okay, there's two nice things about this, is that w has appeared. And this is just some average of u and b. It's not just the average of u times b. There's some sort of annoying shift in there. But it's a constant that we can work out from our experimental data, be it real or in silico. So we know what w is before we start off the analysis. And then f, not only do we know what it is, but we know that it's not positive. So it has constant sign and is occasionally 0. So this means that we have if w is fixed and gamma, the shear rate, is also fixed, then we have an ODE whose sign is constant on the right-hand side. So this is ludicrously simple. If you plot f, by the way, just to convince you that this is always negative, it is always negative. You can show it analytically that it's always negative too. And it has two zeros. One of them is a g of pi, which I haven't told you this, but it corresponds to exactly this point here. And the other one is some other point. It doesn't really matter. It corresponds to this black trajectory, which is somehow between the two. And what you get is this weird cusp point in f. It turns out to capture that properly, you have to do an even more detailed asymptotic analysis to capture it. But also you don't need to, because this is, in terms of this dynamical system, it's just a half stable fixed point. So if I'm doing these dynamics and I'm say moving to the left and I approach here, even though it's 0, any nudge will just continue to move me to the left. So this is unstable in practice. This is quite nice. We have an ODE that's either going to move, we're either going to move to the right on this picture or we're going to move to the left, depending on what W is. That's perfect. So I'm either going to head off to G of pi, which is my minimum, or I'm going to head off to infinity. And as I said, those just correspond exactly to the behaviours that we could see. Going to G of pi is the same as upstream swimming at the centre, and going to infinity is just endless tumbling further and further away. Clearly there are walls there in practice, but we're ignoring them for now. And what's particularly nice then is that what we've been doing is really just tracking the evolution across these contours. So we've got these intermediate oscillations, and what H0 is tracking is how are we moving between the contours on this plot. So either moving toward the center or away from the center, depending on this swimmer-dependent constant. So yeah, we compute this constant, it tells us everything. It tells us what's going to happen eventually. So the proof is then in the pudding. If you trust my asymptotics, you won't care about this plot, but this is just saying the black line is what the asymptotic prediction is for H. And there are very small oscillations going on here that it does track very well. So this is a good sign when you do asymptotics. If your line cuts through the full numerics, then it's worked. If you compare that to the experiment, then, well, we've kind of answered our question. If the W is going to be positive, turns out gamma is positive here, but if our W was positive for this particular swimmer, then we've answered the problem. And it turns out it is. So what we've been able to do is take this model, which you could only really numerically look at without asymptotics, and then reduce it down to, okay, just compute one constant, and then you never have to simulate the dynamical system at all. You can just read off what the behavior is going to be. So this was incredibly surprising that this worked. So it completely explains this experiment, or rather it is commensurate, or it agrees with the result of the experiment. I'm not going to claim we've captured all the physics, but it seems like that enough physics would be these interactions between the changing shape and the changing speed. Notably, if you make any of those constant, it doesn't work. Like, you get none of this complexity, none of the richness that pops out of this. So it really does rely on these intricate interactions that are generating this root omega scaling, which then gives rise to this overall behavior. So really very subtle and some quite neat maths that's going on behind there. And it agrees with the experiment. I'm conscious it is roughly 22, so I'm going to blitz through a very simple problem. And it's very simple. It's quite embarrassingly simple that yesterday and over the weekend, I rushed to throw something on the archive, because as soon as I tell you the problem, you'll be able to solve it in 10 minutes, sort of thing. It's not that exciting. None of you will want to, but just in case. A tiny bit of background fluid mechanics for a microswimmer. The leading order flow field is a force dipole. So you may or may not know what that is, but it looks like this. Microswimming, all problems are force-free. That's essentially saying that the Reynolds number is 0. There's no such thing as inertia, so there must be a total force of 0 acting on the thing. And that means that if you to expand the flow field about the center of this thing, you can't have any contribution from a force, so the next order thing is a force dipole. And the flow looks like this. So for a chlamydomonas that we might call a puller, it pulls in the fluid from the front and actually a little bit from behind, and then it spits it out at the sides. And similarly, if you had a sperm cell that's doing the exact opposite, it's drawing in fluid from the sides and it's pushing it out behind it. So they're propelling themselves in quite different ways. The chlamydomonas drags itself through the fluid, whereas the sperm ejects fluid out behind it. But not in an inertial way. It's quite easy to mix up our intuition for that. And all that we need to take away from this is that this dipole, we often give it, talk about the strength of it, which is assigned strength. And it's negative for a puller and positive for a pusher. There's just some parameter P. Okay, here's our model problem. Suppose we have two of these swimmers, which I've drawn as ellipses, and we've pinned them in place, but we've allowed them to rotate. So we'll call them rollers, because that's what I keep calling them when I talk about this. So we have these rolling things. They're interacting because they're generating dipole flows. You can question whether that's valid or not, but which is an interesting one, but we'll assume it is for now. They generate dipoles, which we'll assume are the same, and they have shapes, which they're changing. So imagine you've pinned your chlamydomonas, they're swimming, they're changing their shape, and they're generating dipole flows, and you're just trying to see how they rotate. So this is a relatively classical model. You can find it in nice textbooks for microswimming, for instance. Look how pretty the equations are, right? Only a few trigonometric terms. So this is very simple. As horrible as it looks like, this is very simple. We have a few parameters. We have the strength of the dipole, their separation, which is constant r, and then we have this b parameter cropping up again, which is their shape. Okay, this looks quite repetitive, and it is quite repetitive. There's all sorts of symmetry in this problem, so it's just the same dynamical system, you just swap the angles because it's all very symmetric. And it doesn't matter what the details of this are at all. If you look at this model and you examine it, it's very easy. You can draw a phase plane for a particular P and a particular B, and you get something that looks like this. It's all nicely periodic, and I've been very lazy in drawing it. But you can write down a phase, or draw a phase plane. And if you look at it for all the different parameters P and B, you get kind of 8 possible portraits. The fact that there are 8 isn't important. The fact that there's more than one is important. But all quite qualitatively different. Here you get proper closed orbits. Here you seem to be moving away from unstable nodes towards stable nodes, and there's all sorts of saddle node bifurcations happening. This is with constant P and constant B, approximating my swimmer in the simplest possible way, constant parameters, which people do, and by people I mean me. I've done this before. However, this is a computational simulation of the flow around a sperm cell, and clearly this is not constant. You'd be amiss if you were to say that this flow was represented by a constant anything. See, clearly the shape is not constant either, it's undulating. So that motivates looking at a problem which you're not at all surprised by. What if you say P and B vary very quickly, just as we've been doing throughout this whole talk? Does it matter if P and B vary a lot? As I say, everybody looks at these models on the left, including me, where you basically implicitly averaged beforehand. I say, I look at my sperm cell, oh, this is a pusher, so P equals a half, say. And I measure its shape and I put in the average. And I see what I get out of the model. It corresponds to one of those phase diagrams. I'm happy. But does it matter? Does it matter if I actually incorporate the fast timescale stuff? So you do the multiscale analysis, which if you've even slightly followed this talk, you could do in genuinely 10 minutes on a piece of paper. So as a challenge, do that, it's quite fun. And what you get out is this leading order system. All the bars are just denoting averages, but the P bars are the same, but something that is different is this final parameter. Instead of an effective shape, the average of B, I now have this weird nonlinear average, and that's coming from the fact that P and B were just being multiplied together at some point in that dynamical system. It's not a deep observation. Everybody knows that if you have the average of a product, it's not going to be the product of the averages necessarily, but it does matter. So in particular, because they're not equal in general, it means that there won't be equality between these two dynamical systems. Okay, that's quite annoying actually. But you do get equality back if you keep any of these two things constant. It's just because the average collapses and you get something very easy. But really, I've been a bit sweeping when I've said that fast dynamics matter. They matter if we want really quantitative agreement with the dynamics, because they're different dynamical systems. It should matter what parameters we put in. But really, the more pertinent question is, does it qualitatively matter which parameters I put in? Okay, as the easiest way to see this is just by one example. If you pick some nice sinusoidal forms for these parameters and you compute what they are, then you can just see what happens really easily. So you just simulate a very, very easy system. Here are the two phase portraits it corresponds to. They're purposefully small because really you just have to look at this plot. If you use the, I'm going to call it naively averaged because I'm calling myself naive in doing this. You just average beforehand, plug it into the model. The eventual configuration of these things is like a T-junction. However, if you use the actual parameters, or what has come out of our systematic analysis, then the configuration is entirely different. They're sort of aligned parallel to one another. So completely different steady state. And notably, this rapidly oscillating black line is what happens if you simulate the full dynamical system. So it does really seem to matter, and your prediction can just be wholly wrong. You can get a huge quantitative difference just by employing what I would have previously said up until a few weeks ago were perfectly reasonable parameter choices.
Quick question in terms of that. So previously, so when you looked at in silico data, the experimental data looked very much like sinusoid.
Yes.
Have you looked at that in this case where, okay, PB bar over P bar is different, but experimentally?
Oh, experimentally very different. So you can measure these things either by doing like micro PAB to measure what the flow is, or by doing something in silico to measure what B is and so on. And you can basically get anything out for your dipole variation. It's well known that a sperm, when it's swimming, sometimes has P as positive and sometimes has P as negative, based on which part of the beat cycle it's in. So you really do get sign changes in P, and the shape can change quite a lot. So in general, one can't expect really anything in particular from this nonlinear average, which is quite sad. It would be nice if you could say, okay, it doesn't matter in this regime. actually, for this particular dynamical system, which is not obvious from what I've shown you, if your P is always of one fixed sign, say it's always positive, it can vary, but it's always positive, then the qualitative behavior will always look the same. But that's not obvious from what I've shown you, and it's a little bit irritating to dive into the details. So there are some regimes where you can be a bit cavalier, but that regime depends on which model you're analyzing. It's not true for all models that if P is always positive, you can get away with just plugging in the naive average. It's different for different models. Something which I only wrote in the archive paper yesterday, which is definitely true, probably. And the take home from this is not that simple models are bad, it's that simple models are bad when you use the wrong parameters. So I still love a good simple model. It's how I develop all my intuition for all this sort of stuff. But this kind of really trivially simple example shows that you should use proper parameterizations, and you can just systematically compute them by doing a simple multiple scale analysis. I'm going to stop talking now. Last slide is, here's everything we've done. Hopefully, I've given you a bit of an overview of multiple scales and how it can actually maybe be useful in a slightly practical context in the second part, where we looked at this experimental data and were able to actually analyze this model and get out a prediction which really does align with experiment. And then also how multiple scales can be a really easy way to annoying your entire community and posing something really simple that actually we can't wave our hands quite as much. But if we do this analysis, which takes you 5 minutes, you can work out exactly what you need to do. And please do ask any questions. If you want any details of what I've talked about, that QR code or my website have links to all the papers and things. But please do ask any questions.
It's also a very nice website.
Okay. Thank you very much. I thank the person whose template I stole. But thanks very much for listening.
Any questions?
Yes, so do you align in their swimming direction?
That's a great question. Yes, they do. The question is, so actually another one of these simple models that I looked at, which is in the archive paper, which is linked, is what if you take one of these dipole models, put it next to a no-slip boundary and see what happens? Do you get the same prediction as you would? So the classical prediction is that yes, a sperm will swim horizontally. And that is true from experiments. It's also true from the simple model, but there's no reason it should be correct from the simple model with the constant parameters that you plug in it. So they do, but it seems to just be serendipitous. The next thing I will do is go and compute these exact functions of time for these swimmers and see, okay, can we get away with it if it's a sperm cell, or can we get away with it if it's
I mean, presumably turbulent bacteria, though, you'd want to be able to...
Exactly. There's a whole range of examples where you shouldn't just be able to be naive and average it. I keep saying naive because it's describing myself. I don't mean to demean anyone. It's just me. I should also give my funder their full title, the Royal Commission for the Exhibition of 1851, which is very long to write in forms, but it does make emails come back to you a little bit more quickly if it's the word royal.
So they're the Crystal Palace people.
Yeah, exactly. Yeah, they built the Crystal Palace, and then all the money that was left over, they invested it and bought Imperial, basically, and all the land around it, which is what funds my work. I'm going to take this as being very thorough.
Yeah, if we don't have any more questions for Ben, we're going to go to the staff room in a second, but yeah, we'll thank the speaker again.